Okay, welcome back. So now we are going to continue with the part 2 metabolism and enzyme, which is in your chapter 4. So before we start the lesson, the outline of today's video, we'll talk about what is intracellular and also extracellular enzyme, and how are the enzymes being produced in the cell and how they are being transported, and what is the lock and key hypothesis for enzyme. So first, intracellular and extracellular enzyme. So enzymes are synthesized in the cell for its own use. So this kind of enzymes, we call it as a intracellular enzyme. For example, like a hexokinase. So remember the ASE nomenclature. ASE means this hexokinase is an enzyme. So the hexokinase is used in the glycolysis, and this is an example of intracellular enzyme which is being used in a cell for cellular respiration. And next, enzymes that are secreted outside the cell are known as extracellular enzymes. For example, like trypsin. Trypsin is a kind of enzyme. It's a special enzyme. Is different as the normal one, which has the ASE uh, at the back of the enzymes. So trypsin, just remember, is a special enzyme that is produced by pancreatic cell. Pancreatic cell is in your pancreas, which is an organ, and then it will be secreted to another organ. So it is produced by pancreatic cell, but it is not used by pancreatic cell. It's being used by other cells in the duodenum, which is a part of the small intestine. So trypsin is a extracellular enzyme. Just remember these two, the two difference of the enzymes. So next, we are going to talk about the production of enzymes. How is the enzymes being produced? So remember all the cellular components here. This is your rough endoplasmic reticulum. This is your nucleus, okay? And this is your smooth endoplasmic reticulum as they have no ribosomes on it. The small blue spherical things is actually the ribosomes. And remember what's the function of ribosome? That's our first step. So first step, ribosome on the rough ER is going to produce protein. And next, it's going to be the ribosome will produce the protein, and the protein will be transported across the rough endoplasmic reticulum, which is the process number 2. And number 3, they will be go to the end of the rough ER, and they will be secreted as a transport vesicle. So transport vesicle is like a car that's going to transport the protein inside. Okay. So remember, at this phase, it's still a protein. It's not being processed or even packaged yet. So it's not an enzyme yet. Just remember, all enzymes are actually protein. Okay. So this transport vesicle will carry the protein produced by the ribosome, and they will go to the Golgi apparatus. Okay, so remember what's the function of the Golgi apparatus is to process and package and transport and secrete out the protein. So in this process, our Golgi apparatus is going to um, process the protein into enzymes. In other case, it might produce the protein into process the protein into like a red blood cell. Red blood cell is a kind of protein as well. Okay, so in this case, it will produce into enzyme, and then it will be packaged in a secretory vesicle. So in this phase, before the Golgi apparatus is a transport vesicle. After the Golgi apparatus, it will become a secretory vesicle. And then it goes to the end of the cell membrane or plasma membrane, and then they will be secreted out. So one question, this enzyme that being secreted, is it intracellular or extracellular enzyme? Okay, because it's produced in the cell, but is secreted out to be used by other cell, so it's known as a extracellular enzyme. Okay. So this is the 1 to 6, the process of the production of enzymes. So ribosomes is the protein of synthesis, and they will enter the rough ER, and then they will become a transport vesicle before going to the Golgi apparatus. Then they go to the Golgi apparatus, and they are being modified, the protein being modified into the enzyme, and they'll be packed into secretory vesicle. And then secretory vesicle, last step, they will go to the plasma membrane, and then they will secrete the extracellular enzyme. Okay, so this is the sixth process that I mentioned before. So next, after done with the production of the enzyme, we are going to move on with the mechanisms of enzyme action. So how is the enzyme is going to work with the substrate for information. So we will use a lock and key hypothesis. I'm sure in your life you know what is a lock and also what is a key. Here's a picture. So we'll use the key to resemble our substrate, okay? And lock is actually our enzyme. So this is the key enter the locks, and we call this as a lock key complex. But as analogy, the substrate will enters into the enzyme at the active site. Do you remember that our enzyme is very specific? Means that this enzyme can only be used by this substrate, okay? So that's by the definition of specific. It's because every enzyme they have a active site. You see the shape? It's only fit with the shape of that specific substrate. So this substrate will enter into the enzyme and then they will become a enzyme substrate complex, okay? Just remember that enzymes, they are specific and they have a active site for the substrate to bind to. So after the substrate enzyme complex, then the enzyme will still remember one of the general characteristic that we talked about it in the last video, that the enzyme will be unchanged and will not be destroyed after the reaction, but the substrate will become a product. Okay, A becomes B plus C, and this process is reversible. So the reaction takes place and the product is being produced, and the product then leaves the active site. And this enzyme is unchanged. And another characteristic is that this enzyme can be reused, is reusable. Okay. So last of this lesson, we're going to learn on this enzyme reaction. So most reaction, any kind of reaction, either, but in biology, we're going to talk about the biochemical processes like cellular respiration or other kind of process. All these reactions in the cell, they require energy, and we call this energy as a activations energy. And usually, normally, without an enzyme, they need a very high activation energy in order to complete the reaction. So activation energy is the energy needed to break the bond in the substrate molecule. So the substrate is actually molecule from the point of view of chemistry. So energy is this activation's energy is needed to break the bond. So in order for us to change the substrate into the product, okay? And the enzyme, remember that the functions of the enzymes is to catalyze, to speed up the process. How it speed up the process is actually by lowering the activation energy. Here's a graph to explain the activation energy with and without enzyme. We'll have a look on this. So you see the x-axis is the reactions, okay, any reactions in the cell, maybe a cellular respiration, and the energy will be the y-axis. So you see the red line is the process without an enzyme. So a process without an enzyme, they will use a higher energy. They will require or they need higher energy to complete the reactions to produce the products. However, with these blue lines you see here, with enzyme, actually the process will need a lesser energy, you see. This is the difference of the activation's energy. And the blue one, with enzyme, they require lesser energy to produce the product, to complete the reaction. So this is the function of enzyme, okay? So enzyme is very important in our body. So let's recap for the whole video. Today we learn on what is intracellular and what is extracellular enzyme. So intracellular is the one that produced in the cell and used by the cell. Extracellular enzyme is the one that being produced by the cell and being secreted out and go to other cell and being used by other cell. And the enzymes, we also learn on how the enzymes is produced and also how is it transported. Remember the 6 processes. And we also discuss on how what is the lock and key hypothesis for enzyme. So lock will resemble as our enzymes, and key will resemble as our substrate, okay? And they will form a substrate enzyme complex. And then after the reactions is finished, a product will form and the enzyme will remain unchanged. And we also talk about what is the activation's energy, and the presence of enzyme is actually to function to lower the activation's energy, so to speed up the whole process, for example, like cellular respiration in our cell. So that's all for today's video. Thank you very much for watching. I will see you in the next video. Bye bye.